So this work on the leaf-footed bug is done in collaboration with Houston Wilson at UC Riverside and Jocelyn Miller at UC Riverside. Jocelyn's the great pheromone chemist who's done so much work with so many different insects. And Houston is a relatively new entomologist based at the Kearney Agricultural Center. But I'm going to throw in some background work that I did on these insect pests with Glenn Yakota and Judith Stahl as well. So as an outline, we'll just talk about the bug species. I want to put in context leaf-footed bug compared to the stink bugs in terms of damage, in terms of monitoring. Then we're going to look at the economic injury of leaf-footed bug, and I'm going to point out why I think it's that overwintering population which is so important. And because the overwintering population is so important, that's what makes monitoring so important, because this can come in 1 year very low numbers, another year very high numbers. And I remember a couple of times when we had just super populations of leaf-footed bugs and everyone started to spray. And then people kept that spray up for 3 or 4 years, even when the densities were relatively low. So to monitor that overwintering population coming into your orchard is key. I'll discuss a little bit about the seasonal biology, why there are these superfoods, such as pomegranate, which allow the population to increase in size. And then we'll get to the new pheromone monitoring of leaf-footed bug and where we stand, and how far away I think we are from a commercial program, and then put that into context with we're still going to be using the beating tray because we still have got the stink bugs out there. So just as a background, kind of comparing leaf-footed bug to the stink bugs are just really colorful creatures as nymphal stages. So we've got the eggs in the upper left, and we've got a cluster of, that's the green stink bug. This bug is not green until later in the stages. And if you've ever seen the egg mass and looked at the first instar of stink bugs coming out, you see that they remain kind of clumped for a while. In fact, for many stink bugs, the first instar is not a feeding stage. They're just kind of hanging out together, then they disperse and start to feed. So this is our Chenevia. It used to be Acrosternum. Common name is green stink bug. We'll go through a couple of stages. It starts off first instar, upper left, going kind of clockwise. We now have got a second instar, third instar, down the lower left, fourth instar, and then as a fifth instar, it starts to look like a green stink bug. So one of the differences between a leaf-footed bug and the different stink bugs is that I can see a leaf-footed bug nymph on the beating tray and know immediately that's a leaf-footed bug. I drop a stink bug nymph on that beating tray, and there are so many different color forms for the 5 different species, actually 6 different species, if you include Brachyphylla, which is the good one, that it's hard to know which stink bug species we're dealing with. I think that damage is also important. Damage is one of the key monitoring tools early in the season to find out if you've got one of these large bug pests. And one of the issues for me, and this is the adult green stink bug, we just saw all of its larval stages, nymphal stages. One of the things for me is that if I see this damage in the field, I can't tell you what stink bug species caused this damage, or maybe a leaf-footed bug species. So when you are looking at damaged nuts, you just know it's a large bug, and sometimes early in the season, drop nuts can happen with the small bugs as well, such as Calochorus and Lygus. And if you look at the 2 pictures up and down, the almond damage, it's got that little gummosis signal. That's always not a signal of a large bug feeding. Sometimes it's just this gummosis coming out, a physiological flat tire-like event. What I like to do is to cut them open and follow that gummosis, that line from the stylet, from the mouth parts of the stink bug or the leaf-footed bug, to that kernel to see if we're getting that brown spot starting to form. Now, I mentioned the damage between the stink bugs and leaf-footed bugs can be very similar. The nice thing about the leaf-footed bug is that to me, it looks like a leaf-footed bug as a first instar, second instar, third, fourth. It does change color, but the shape remains the same. So in that top picture, we've got the egg mass of the leaf-footed bug, and we've got the 3 little leaf-footed bug first instars pushing their head out of that little round circle on the egg mass and starting to emerge. Like the stink bugs, those first instars tend to emerge from one egg cluster at the same time. And they hang around that egg cluster for a little while, but they are a feeding stage, unlike the stink bugs. They will start to disperse as a first instar and start to feed. That middle picture is about a third instar leaf-footed bug. They're actually very colorful. They're kind of red and black at that stage. And that lower picture is now a 5th instar leaf-footed bug, and it's starting to look like the adult leaf-footed bug with the pads coming out. But I wouldn't mistake this for much of anything else. You might look at something like an immature assassin bug and think, oh, that looks like a stink bug. But once you get to a little bit more experience, you'll know leaf-footed bug is a leaf-footed bug, assassin's an assassin bug. One of the other differences I have with stink bugs versus leaf-footed bugs, and I've talked about this before at Pistachio and Almond Day, the amount of damage caused is really related to size. So the first instar causes less than the third instar than the adult. It also means that the smaller stink bug, like Thianta, red-shouldered stink bug, causes less damage than the adult leaf-footed bug. The adult leaf-footed bug is just larger, and it's got larger, stronger, longer mouth parts. That means it can feed a little bit later into the season and still cause some damage. We see a pistachio above and an almond below. You can follow actually when you see the kernel necrosis on the pistachio, if you look closely, you can see where the mouth parts went. And of course, these can also import diseases into the almond, and that's what the bottom picture shows. When I started this work with Dick Rice and Walt Bentley, we talked about 2 leaf-footed bug species, Leptoglossus clipialis and Leptoglossus occidentalis. We rarely saw occidentalis in the almond field, but clipialis, the one with that long nose, was the common leaf-footed bug species. Things have changed. We now have got this Leptoglossus zonatus. That's the one that has these really large overwintering populations that you see in all kinds of overwintering habitats. We don't know why it changed, if it had to do with climate change, if it had to do with warmer winters, or it just migrated and moved. We get invasive species all the time. So to kind of highlight the damage and how much damage these can do, we did these feeding studies. We put cages out. We put one nymph or adult leaf-footed bug into the cage, and we gave it a 7-day feeding period at different seasonal periods on different almond cultivars, and we rated the different types of damage. So here's where I think it becomes really important, especially for the almond growers, not as much for the pistachio growers, because of compensation of the fruit yield. So this is one adult leaf-footed bug in a cage for 7 days, and it dropped 95% of the nuts in that cage. Tremendous number compared to the control. That's when you get a large population, you're walking the field, and you see all these little BB nuts down on the ground. You can't do anything about it. That could have happened in a 1-week period if there was a huge population overwintering somewhere nearby. That's why monitoring becomes so important. Later in the season, when we get fruit set, we're looking at dropped nuts, damage to the shell, and damage to the nut. So this is still a fairly soft almond. We're getting still a lot of dropped nuts, and notice it does vary a little bit amongst cultivar. We're getting shell damage, but now we're getting a little bit less nut damage. They're poking, they're feeding, they're not getting to the kernel all the time. And you can see that this really increases as we progress through the season. So by June or July, we're getting very few dropped nuts at this point in time. So the nut damage you get goes to harvest. We're still getting damage to the shell, that gummosis coming out, but now we're not getting as much damage to the nut itself. Stink bugs and leaf-footed bugs are basically seed feeders, but they can get moisture from all kinds of things, feeding on citrus leaves, things like that. So as the season progresses, you get less damage from the leaf-footed bug and the stink bugs. And if you end up with first, second, third instars, you're in between generations, at this point in time, they really can't penetrate the shell to the kernel and do very much damage at all. Seasonal biology. This is all important to understand why monitoring is important. So leaf-footed bug overwinters, we'll see next as an adult. You got these aggregations someplace near your orchard. They tend to hit almonds first, then they go into pistachios, then they go into pomegranates. But to say leaf-footed bug is a pest of our tree crops is missing the point that these things are feeding on all kinds of different plants. So in between these different crop systems, they can feed on all kinds of weedy vegetation, anything with a seed, to hold them over, to have them migrate from one field to another. I know with stink bugs, we found them on Russian thistle like crazy during the early springtime. This is also why sometimes we get a season with a lot of rainfall, followed by a spring with a lot of weeds, followed by a large healthy population of leaf-footed bug or stink bug. This is work Houston did with a Fresno State graduate student, and it's going to be difficult in the back to read that, so I'll just tell you what it says. They're basically doing, you can see this dissection of a leaf-footed bug. They're looking at gut analysis. They're using a molecular tool to look to see what's inside the gut of leaf-footed bugs. And they're collecting leaf-footed bugs from almonds, pistachios, and pomegranates. And what they're finding is that, yeah, for example, in pistachios, about 35% of the gut content was pistachio. But we can see in pistachio, squash, cucurbita, also had a lot of leaf-footed bug signal. So 32% of those leaf-footed bugs in pistachio had fed on squash at some point in time. Same thing with almonds. Most of the leaf-footed bugs in almonds fed on almonds, about 23%. But about 36% also fed on pistachios. This is just to tell you that these insects are moving around. Remember, the nymphs are not a dispersal phase. They don't have wings that work. They have wing pads. So this is all from the adult or a nymph becoming an adult and then moving to a different site. So monitoring is important because these are migratory insects. They're moving all the time, even within one generation. This is work I did years ago when Brent was still young. So we're looking at top graph egg strands, middle graph nymphs, bottom graph adults. We can see February, March, April, May, June, July, August, and so on. Second generation, so we've got that first generation from the overwintering population. Second generation is coming out May, June, and July. Eggs, nymphs, adults. Third generation, July, August, September. And going into the adults, that's the overwintering stage. In most areas, we don't get a fourth generation coming out. On pomegranates, I think we do get a fourth or partial fourth generation because it's such a good food source. Typically what happens, and I'll show you a slide later, by November, if you haven't made it to the adult, a dispersal phase where you get to a good overwintering site, you die off. In pomegranates, they seem to all make it to the adult. Where are these overwintering sites? They're everywhere, any place with shelter. Palm trees, eucalyptus trees, wood piles, citrus, olives. We find them all over wherever there's shelter, and they need to feed a little bit to get some moisture during this time. It doesn't have to be on a nut. It can be on any source with a little bit of moisture. One of the trials I did back in the day is we looked at leaf-footed bug adults, medium-sized nymphs, and small nymphs as overwintering animals. And we can just see here in red, adults with food survived until April when they're moving into your field. Adults without food, about 20% survived and made it into your orchard. But if you were in December a small nymph or a large nymph, you died out. You couldn't make it through the winter. So that's why the overwintering adult stage becomes so important. That's the stage that makes it into your field in the springtime. And lastly, before we get to this monitoring portion, and stop me if I go too far. So what we did is we dissected that upper right graph. You see the ovaries and the oviducts of an immature ovary in an adult leaf-footed bug. Down below, you see those barrel-like brown objects. That looks just like the egg mass. That is an egg mass inside the oviduct. What that graph shows is that at the end of January, the females had immature ovaries. The ovaries were not developed. They developed their ovaries between January, February, and March. And so in April, when they're going into your orchard, they've got eggs ready to deposit. So this is something we have to consider when we start to consider pheromones for monitoring, because we found that they're actually mating in their overwintering aggregation. So if we're using a sex pheromone to attract them into the trap, maybe it will or will not work as well early in the season when it's so important to monitor. So with that as kind of a too long background, let's get into monitoring with pheromones. Where are we at? And thank you to the almond and pistachio boards for funding this research. So we used to look at different kinds of monitoring programs, the beating tray, the sweep net. We even used a DVAC, and with Bob Beatty, we did an insecticide spray to look at all the insects inside the pistachio orchard. What we found was that the beating tray was still the best for large bugs, stink bugs and leaf-footed bugs. Sweep net was the best for small bugs, Phytochorus, Calochorus, and Lygus. So first you can go in while you're using the beating tray, and you can visually look for damaged nuts on the tree or drop nuts on the ground. Do this, I would say, in April and into May. You can use a beating tray at that same period. Typically works a little bit better early in the morning when it's a little bit colder. The insects tend not to fly away. Hold the tray below the canopy, couple of strikes, look and count to see nymphs or adults. That's fantastic, but again, we're worried about this migratory population early in the spring. And I know if I've gone up and seen leaf-footed bugs fly away from me as I'm approaching that tree, and they don't make it onto the beating tray. I actually like just using a 10-foot bamboo stick for a leaf-footed bug and walking through the orchard and whacking the tree, and then watching the leaf-footed bugs fly away because they've got a distinctive sound as they leave. So I thought that worked for me as well as the beating tray, but it doesn't work as well for the stink bugs. So the beating tray, the bamboo stick, that's an active, time-intensive sampling program. And there's not a great correlation between what you count and the amount of damage you get. And again, you have to be in the orchard to take those counts. So in all these other things like the navel orangeworm, we've got these lures that pull something into a pheromone trap, a passive trampling program. So I started this with Jocelyn. Houston was hired. He took over the field portion of it. Where are we now? So we know the leaf-footed bug has got an alarm, an aggregation, a sex pheromone, and an aphrodisiac. The males put out the sex pheromone, and they also use an aphrodisiac to allow to kind of coerce the female to allow mating to occur. So the idea was, can we use this to develop a monitoring program? First thing we had to do was to find out what traps will work. So this is without a pheromone lure. We looked at small, large pyramid traps, the brown marmoted stink bug, clear sticky trap, hanging paddle, and a uni trap. And what we found was that hanging panel trap was the best for leaf-footed bug. We looked at with and without fluon. We found that we had to have fluon added to the trap to increase our capture. That increases the labor in putting these traps out as well. And we looked at trap color, and we found that yellow and blue had more captures than the other trap colors, red, black, white, and green. So we know what traps work, we know what color of traps work. Now we needed a lure to pull them into the traps. What Jocelyn does is he takes a male, captures the pheromone, pushes that pheromone through a machine that's got a female attached with wires to her antenna, and uses a GC, a gas chromatograph, to look at the chemicals going across that female's antenna. And when she vibrated, when she showed excitement, he looks at what chemical was there. And they discovered a new chemical to science called leptotrene. So the next phase was to test this in the field, and that's what Houston and I did, Houston for the most part. And we've got 3 different almond sites, and you can see it was the same pattern in each site. There was a reaction, more leaf-footed bugs captured, mostly females, but some males, with leptotrene. And then we also had a combination of leptotrene with 2 other components. We want it to be simple because the more components you have in a pheromone, the more it costs to make. So we wanted to keep the cost down, and we got a reaction, a collection of insects with just leptotrene. Lepto because of leptoglossus. That's why they named it that. So, concluding remarks, where are we? Stink bugs, leaf-footed bugs cause the same damage. They look different, but they cause the same damage. One of the questions I've got about a monitoring system is once we start monitoring with pheromones for leaf-footed bug, we can't drop monitoring with the bee tray for stink bugs. And stink bugs, 4 different species, so we have to worry about maybe 5 of BMSB becomes more important. The monitoring period that's really important is the overwintering population. And we still don't know how much attraction we're going to get from this pheromone during that important overwintering period because we showed that they can mate in their aggregation during the winter. Moving towards this lure, though, we have found a suitable trap. We've identified the key pheromone components. Jocelyn's able to synthesize all of those components, and field trials show that we can put this lure out, leptotrene, and collect the female mealybug. So what we want to do now is to optimize the blend. In Europe, they're getting thousands of leptoglossus per trap per week with the different species. You saw our numbers, we're getting 3, 4, 5. Sometimes the blend of the material is really important, like the blend of gasoline for a high-performance vehicle. So we're going to test that out this year to see if we can't collect a lot more, improve the cost of synthesis of these compounds to help with commercialization, and we're going to start comparing trap counts to orchard damage to see if there is actually a correlation that makes sense to use these traps and lures. So we're still a ways out, but we're getting closer. With that, I thank you, and I think I'll take my questions now. Yeah, Emily. Yeah, so the question was, is the blend we've got right now aggregation or sex pheromone in nature? We really don't know exactly. We know that there was a reaction to it. And one of the things we're going to try this year is with BMSB, they've got a kill strip in there too, right? So one of the things we're worried about is that this could have been a aggregation or sex pheromone. And Jocelyn thinks it might be used for both, but they were going to the trap, dropping in the trap onto this sticky part, and then they were sending out an alarm pheromone saying, whoa, I'm here in the trap dying, don't. So we're going to try to do some kill strips this year to see if that helps a little bit. But we don't know yet if it's an aggregation or a sex pheromone. Yeah, so the question, the comment is, the observation is that Aldrich, it was Aldrich and Carmel, okay, and they're getting more of the stink bugs. My question to you, is it the green stink bug that I showed? Yeah, so the green stink bug is one that it more commonly than the other stink bugs overwinters in the orchard. You go out there on a nice warm February day and you can start to see them going up the trunk a little bit. So it might be a population which is resident there. And I think they do like, I don't know those cultivars, are those softer shell cultivars? Yeah, they like the softer shell cultivars typically. So what I do is make sure that it's the green stink bug and then consider doing maybe a post-harvest, something to knock that population out. The other stink bug species tend to be a little bit more migratory, especially the red-shouldered stink bug. How far will these leaf-footed bugs, we'll focus that on migrate? Not as much as the navel orangeworm, but quarter mile is a guess from an overwintering site. And remember, each generation, you get another chance for movement. It's the adult population. You've got 3 generations per year, so they can go miles over the 3 generations. Well, it depends on what you're spraying. So most of the pyrethroids are what we use for the stink bugs. I know that people are trying to move away from pyrethroids with NOW because of resistance issues. But that's been our best tool. I know Chris Tollerup did some work and suggested belay, which is a neonic, also had control. There's, to be perfectly honest, the PCAs in this room have got more experience than I do with what's working for stink bugs and leaf-footed bugs with insecticides. We put out different crushed up almond meal and put them in bags, and what we found is that you get some egg masses on there, and then what we're seeing when we see the nymphs are the production from the egg masses. So it works a little bit like an NOW egg trap, I think. Yeah, good observation. Thank you.
All right, so next is going to be my talk, and I'm going to be focusing on management tactics in other crops. So my name is Fann. I am the new pest management specialist for the almond board. I started in September, so I haven't met many people, but this is a great opportunity. I recently finished my PhD in entomology from the University of Kentucky, where I studied the brown marmorated stink bug and looked at management practices in organic and small acreage vegetable systems. So while I am new to the almond industry, I am familiar with the brown marmorated stink bug and management tactics for this pest. And so what I'm hoping is with this talk is it's not going to be focused on almonds because we don't have a lot of research focused on BMSB or the brown marmorated stink bug research projects for BMSB, but I'm hoping that this will help to inspire some future research projects and also give you all some ideas about what you might be able to do to help monitor populations in your fields, but also a lot of the work that I did during my PhD was grower inspired. So there's a lot of minds in the room who are in the field that can help researchers gain ideas and inspirations for research projects. And so I just want to emphasize that because it's important. So first, I wanted to talk about why stink bugs are a problem, or specifically the brown marmorated. So before the introduction of the brown marmorated stink bugs were primarily seen as sporadic and secondary pests. They weren't, and they often didn't cause much concern for growers. I know that's been saying with leaf-footed bugs it has been a problem, but brown marmorated's now becoming an increasing concern, and we are aware. But with BMSB, it has a very broad host range. It has over 200 known host plants. This can go anywhere from tomatoes, peppers, sweet corn, almonds, and then also feed along the fields of lots of weedy areas as well. And so this broad host range allows it to switch between hosts as they become susceptible throughout the season. Also, they have a very high mobility. The brown marmorated stink bug nymphs, so they're those smaller instars, they can travel long distances. They crawl very far, and then the adults are very strong flyers, so they can move long distances to get from host to host. Another thing that's important is that every life stage beyond the egg has the potential to cause damage. So this means that from the point of hatching, after they move off that egg mass, you can begin to have damage in your orchard very quickly, and for its entire lifespan. So it makes it very difficult to control once you have most life stages causing damage. Another thing is that they cause direct damage to the crop. So like we've seen in other presentations, it does cause that brown spots on the nut, And in other crops, it causes stippling and corking, and so I'll show that in some other photos. But because it is a direct pest, it is difficult. But also, it is difficult to control due to the mouth parts. So, unlike something that chews, the stink bugs have piercing, sucking mouth parts. So, unless they're getting some direct exposure, oftentimes they're not picking up enough chemicals to kill them. So, it can be difficult to control that way. So what I'm going to be talking about is 3 different management practices to help. So looking at monitoring, following after Dr. Daane, how monitoring is very important for understanding what's in your field, but also because monitoring tools for BMSB, there's some great tools available. Looking at biological controls, so what might already be in your fields and what you might want to preserve that's there. And then one management strategy that I conducted a research experiment on in sweet corn, but I think this could transfer over into an orchard system, is using an attract and kill strategy. And so this is where I'm hoping you all will have some inspiration or some questions to let me know or let us know if this will be a good idea for almond production. So talking about monitoring for the brown marmorated stink bug, we have an aggregation pheromone lure for BMSB. And so this aggregation pheromone lure, it attracts males, females, and nymphs. So it attracts all those mobile life stages that are causing damage to your crop, which is really important and very helpful. This aggregation pheromone lure, it has a plume range. So if you have it in a location, the plume range is about 50 feet or 50 meter on either edges or either edge. So you have this surrounding area that you would have this attraction phase or attraction to this lure. And then it can also attract some other species of stink bugs, but it doesn't do it very well. So this is not good for other species. It is very good for brown marmorated stink bug. And it's because it's a dual lure. It can attract some green stink bugs, some browns, some rice stink bugs, red shoulder, and harlequin. But again, these populations that are caught on the traps are not, or these numbers are not very high. It's strategic for BMSB. And then also the monitoring tool can be used all season long. So during my research, I was setting them out in March and being able to see those overwintering populations emerge and catching those in the traps early, and then seeing that throughout the season until September when they're going back into overwintering. We have the pheromone lure. It's the dual lure. We have these 2 little black strips. You set it up on a double-sided sticky panel, and then it could be placed on a wooden stake or a post. Don't see many wooden posts around here or fences around almond orchards, but a wooden stake works well. This is what it looks like in the fields where you would want to place it not too close to your orchard because it is an aggregation pheromone, which means it's going to attract insects, it's going to attract insects to that spot, and so you could potentially increase the damage to the adjacent trees. So you want to place it like along the area where you're driving, so like the road or something where it's close to your orchard, but it's not going to increase the damage. My other suggestion would be to give it to your neighbor, and you could have your neighbor get all of the stink bugs that you don't want. But if not, I'm just kidding, you can just put it on your dry rows. So I'm looking at some biological controls that have been evaluated for the egg stage, but also for nymphs and adults. What's nice about BMSB nymphs is they all look very similar, except for the first instar. They're red, but they're pretty similar that you can see these in the fields when they're there. Looking at the biological control agents for eggs, there are many different types of biological control agents. We have some chewing predators, and so things that are unlikely, like grasshoppers, katydids, spiders, have been seen to be very helpful and beneficial biological control agents for stink bug egg masses. And then there's also some piercing sucking insects that help control populations, some other stink bugs, spiders, and damsel bugs. And then we have some parasitoids that are some natives and also some introduced parasitoids. So this means that they're laying their eggs inside the eggs of the stink bugs. However, with all of the research kind of that has been conducted, the best thing that we were seeing using sentinel egg masses, so egg masses that were placed out into the field, we were seeing that chewing and sucking predation was higher than the parasitoids that were killing egg masses. So we were getting anywhere between 30 to 70% of egg masses being killed with chewing and piercing and sucking predators versus anywhere between 2 to 3% with parasitism. So the parasitism rates are quite low. So you want to have those grasshoppers and spiders in your orchards. Also, when you're managing the pest at this egg stage, this is, again, the only stage that isn't causing damage. So trying to stop the problem before it happens is a great way to look at this. Another thing is looking at the nymphal stages in the adults, and so there are fewer predators at this stage because they are pretty hardy and they do have that chemical component that makes them not really tolerable. Or tasty. So we have some other piercing and sucking predators, and then also some parasitoids like some flies that can lay their eggs on the nymphs and adults and then kill them that way. So this is where I wanted to spend more of my time in looking at the attract and kill traps. And so a little bit of a background story here about how this trap got started. So in Pennsylvania, there was a peach grower who had a U-pick, and he was one of the first ones that got hit with BMSB. And his orchards were getting hit or had such high populations that he was having to have his farm hands go out and pick the peaches before he opened up for U-pick. So in sheer desperation, he had some old netting that he dipped in some insecticide, put it on a hook, and baited it with those aggregation pheromone lures and put them into his wooded tree line. And so he was just trying to do anything in order to try to reduce those populations in his orchards. And so when he did this, he was killing anywhere between 400 to 600 stink bugs a week using this method. And so he was attracting the stink bugs to the netting. The insects were landing if they were adults or crawling up if they were nymphs, and they were getting that dose of insecticide and dying. And so he showed this to one of the researchers in Pennsylvania, who showed it to kind of the work group, and that's where this next research I'm going to be talking about, the inspiration came from. They start off as called the ghost trap because it looks kind of ghostly in the wind with that white sheet cloth, but there's different variations, and you can see the different variations that researchers have looked at. They've looked at different panels, they've looked at bunching it, and kind of different methods. So it doesn't have to be just a specific standard, it's the concept of the idea. So for the research that I did was looking at attract and kill strategy, is that it's attracts targeted pests to a designated area. So in this case, I'm trying to pull those stink bugs to a location and have them land on this insecticide impregnated netting to be killed. It needs the appropriate stimulation to get the targeted insect pest. And then also using a successful attract and kill strategy. There can be success with using it. We've seen with other growers and peaches that it was helpful. But then with insects concerns, so where I was in Kentucky, sweet corn growers were starting to get hit really hard. Some of the growers were losing 100% of their crop due to stink bug feeding damage. So that's why this work was focused on sweet corn. But like I said, this is just to give you all an idea of how you may be able to do something new in your fields or in your orchards. So this is the type of trap that I set up. I had PVC pipes where I baited it with an aggregation pheromone lure and then draped insecticide impregnated netting around it and bunched it up at the bottom, because my thought was I want to capture those migrating insects from the nymphs to crawl up and get as much insecticide as possible, which research for this, I was able to see that they only needed 5 seconds of exposure to be killed within 24 hours. So it doesn't take very long. And then since I wanted to make sure to target adults and nymphs, and I wanted to focus that instinct on them climbing up. So that's this trap design and method. So we set it up around a cornfield plot. This was done over 3 years' time, but I'm only going to show 2 years. The first year that we did it, I put the traps too close to the field and ended up causing an increased amount of feeding damage to my sweet corn around the borders. So that didn't work. So the next year, I pulled them back and I placed the traps surrounding a sweet corn field, and I placed them about 15, oh man, I can't remember the math, so about 15.2 meters away from the field, and surrounded the sweet corn field, and then took samples, checked the traps, and did the analysis. So here is a sweet corn field, and you can see the traps. There were the 8. This is 4 of them. You can see pretty clearly surrounding the field. And so doing direct counts, we were able, and so these numbers are kind of low, but we could see there was a significant difference in fields that had an attract and kill around the sweet corn field versus the control. So there were fewer stink bugs that we saw in cornfields using the attract and kill traps than we did with control. When we look at the ear evaluations, the average damaged kernels per 20 ears, we had fewer damaged kernels in the treatment versus the control. And then when we looked at the average of unmarketable ears, the same thing, the attract and kill plot had fewer unmarketable ears. This is suggesting that this is what was the attract and kill strategy was working. So the takeaway from this I wanted to say was that attract and kill traps placed too close can cause increased damage, but when they're spaced properly or kind of further away, it can reduce the damage. Also, we have that it can reduce pest populations in the field. It can draw in stink bugs from far distances. The trap itself is relatively inexpensive to make when you can use things you have around the farm to make that stand and stuff, and you don't need a base to capture. You're not really interested in looking at the numbers and stuff at the base. And then one of the thoughts that I had is you could potentially combine this with other lures. So I had tried this with a Japanese beetle lure, which I know you guys don't really have a problem with them here, and with a stink bug lure, and it didn't seem that these were competing with each other. So maybe there's a potential to use this for multiple stink bug species or something of a similar family. And then a bigger thing is it could possibly be scaled up to farm scale, which is where I'm reaching out or talking to you all to see how maybe this could be expanded beyond a small field trial. For wrapping up, monitoring for stink bugs can be helpful in determining populations are present in the field as a season-long monitoring tool. So you can use it from March through September. Also, biological controls in your field are important to see what's there and understanding what you already have and not trying to kill them off. But also looking at maybe another research management practice is looking at attract and kill strategies. So where you might be helping to kill populations on the borders of your fields and not letting them get into your orchard and causing that damage. So I have lots of people to thank, but I am open for questions. You don't need to place the traps very closely together, so I can't give you a number exactly, but the distances we were spacing them, when we were doing regular population monitoring is 50 meters away from each other, so you can space them out pretty far. The material, it was a deltamethrin insecticide impregnated netting. So the company that creates it quit selling to the U.S. for a while. So I think there was a couple of researchers who were working with BMSB who was looking at trying to make sure to get that back on the market in the U.S. and I'm not sure what the status is on that yet. The original one that the grower is using is the shade cloth that they put over their peaches and or their berries. Yeah, so they used some of that initially, and they just yeah, and they had dipped it in a pyrethroid. No, we didn't see that a color of a fabric mattered exactly, but with the black one, I think that like we had looked at using the black because it's not attractive to pollinators or anything in black. It was very targeted towards stink bugs. Yeah, and there I think in the 3 years I did the study, I killed a total of 10 bees. Like, and that's because I set out like 36 traps a year. That is the blurred line question that I'm not even sure about because it's not in the field, but I don't know about that if it's legal to use in organic setting.